Oh. Yeah, so cyanobacteria are probably arguably one of the most important organisms that have evolved in our planet. That's because they were the first prokaryotes to evolve oxygenic photosynthesis. So this is the photosynthesis that most people are used to. They fix carbon dioxide, they use water as the electron donor, and they make sugars. In that process, they release oxygen as the electron donor. And I think it's sort of acceptable to say that they have transformed the geochemistry of our planet and the biology of our planet. The geochemistry, because they have oxygenated the oceans, the atmosphere, and the biology, because they've been involved in a lot of different symbiotic events, including the chloroplast, which led to photosynthetic eukaryotes. So, and what you get through time is this interaction between obviously biology and physical processes. So they would have been fundamental as the Earth became more and more habitable. In addition to oxygenic photosynthesis, because they are also able to fix nitrogen, not all of them, but they transform atmospheric nitrogen into bioavailable nitrogen. These two metabolisms would have been sort of fundamental as these organisms start engineer their environment during the early Earth. Here I have a diagram showing primary productivity through geological time. It is a cartoon, and we have geological time here at the bottom with past to the right, the Archean, and present to the left. These organisms are primary producers and are representing the biological pump. In order to understand how oxygen has accumulated through time, we need to have this idea that there is some carbon burial. So we have organic carbon going into the seabed. This organic carbon gets buried, and then there is a differential between photosynthesis and respiration. And you'll sort of see when we need to understand, because it's not just about cyanobacteria pumping oxygen, we need to think about this carbon burial. One of the things that I've been really key to do during my career is really understanding how some of these evolutionary events in cyanobacteria might have contributed to some of the things that we see in the geological record, and hopefully this will become a bit more clear as we go along. It's easier to get an idea of what primary producers are doing in the last 500 million years, because we have a better geological record, but also because the primary producers leave a better record, calcification, things like that. This is much harder to see when it comes to the Precambrian, and most of the research that I'm interested in doing really concentrates in this time period. And in summary, the questions that I've been keen to understand are: when photosynthesis evolved, when we have the earliest cyanobacteria, and when they became ecologically dominant, and in particular, both in land and in the ocean. I think partly because I get the impression that when it comes to sort of geochemistry, and obviously those are complicated concepts and subjects to understand, they have a naive idea about cyanobacteria, and cyanobacteria are hugely diverse in lots of different habitats. So we can't really grab one cyanobacteria from a lake and extrapolate things about the early Earth. But there is definitely, we can really link the evolution with the geological record. So I've been keen on that. On that subject, and because obviously primary producers are so important, the idea of the origin of these organisms has been approached by different fields. From here, for instance, I'm illustrating the sulfur isotopic record, where they imply origin of, or at least what cyanobacteria were doing. The sulfur isotopic record is being fundamental in sort of dating that the great oxygenation event happening around 2.5 billion. So, you can approach it from the geochemistry. People have thought about it from the fossil record. And I should highlight here, cyanobacteria are one of the prokaryotes that have left some of the best fossil record. So you can have a cyanobacteria at 800 million years ago looking exactly the same as something that we have today. People have used biomarkers like the 2-methylhopanoids to date cyanobacteria. It has its issues and it's not used anymore, but so different fields have attempted to say something about the origin. Same when it comes to the biochemistry of the reaction center proteins, in terms of the timing of the oxygenic and non-oxygenic ones. And here I have phylogeny. Those are sort of the tools that I've used to understand cyanobacteria. Now, when I first started, this is kind of part of my literature review back when I first started with understanding the evolution of cyanobacteria. And this is a paper, very old now, and then they have 16S, which is good at establishing relationships of cyanobacteria that are relatively close related, but you can sort of see here that this tree is like a fan. There is no, the deep branching relationships are not well defined. So my first paper on cyanobacteria attempted to do that, to understand those deep branching relationships. And it's sort of the first attempt to use genomic data. I can't remember exactly, maybe we had like 13 genomes at the time. But what we really wanted to do is understand what appear first and, gradually when things appear. And partly because you can then start testing hypotheses like, we can then look at trait evolution, see if the first cyanobacteria, say, were unicellular or in which habitat they were found, things of that sort. So that was sort of the attempt to sort of understand directionality and sort of when different traits appeared. In terms of the methods that we use, we do a lot of sort of either look at genome, we sequence genomes, we look at data that has already been published, but we do phylogenomics. So lots of different genes to understand the phylogeny, the relationships, a lot of molecular clock analysis. I mentioned how they have a good fossil record. So, and I one of the sort of people who want to do use the microfossils, and a lot of other techniques is trait evolution. There are lots of different methods, but most recently we've been using stochastic mapping. You can have traits that organisms have today, and then you can estimate what an ancestor was doing or looked like with a particular sort of some statistics and support. So those are the things. So whenever I think about when I'm going to be talking about things, many times there's so many different layers of these different types of analysis. We also use comparative genomics. We look at sort of how maybe gene clusters are arranged and maybe specific for a sort of pigments and things like that. and also think about sort of testing for lateral gene transfer. Or often what I find is when people have said, oh, here there is some lateral gene transfer, what often happens is that maybe there was an ancestral duplication event, and then different lineages evolve different sets of these genes. So maybe something would have, kept 2 copies, maybe other ones kept only 1. But then if you look at lots of just this gene phylogeny, it's really hard to tease apart, and people just immediately say lateral gene transfer. So I'm completely obsessed of sort of cannot really tell apart lateral gene transfer, because it just tells you, well, I think it's interesting from the evolutionary perspective. Now, to summarize, and many different papers, this is something that potentially is of interest. Is it's a review paper in Trends Microbiology of this year, and it sort of summarizes kind of like the last 10 years of the work that I've done. Here we have, again, the geological record at the bottom. This time we have past to the left and present. Here we have the profile of oxygen through time, the great oxygenation event, the first one, which wasn't that great, in a way. Oxygen levels remain relatively low for most of the time, the Precambrian, not, probably, to the most sort of 1% of today's oxygen concentrations. So it really, the oceans started to become oxygenated at the end of the Precambrian, and this is known as the Neoproterozoic oxygenation event between 800, 600 million years ago. Now, to cut a very long story short, what we know from the phylogenies and doing all these different types of analysis is that the early cyanobacteria were unicellular. Filamentous forms evolved relatively soon after. There is a radiation of this. So we have here an ancestor called, I called it the macrocyanobacteria and microcyanobacteria. But if you look at a phylogeny, these 2 ancestors led to the biggest diversity of cyanobacteria that we have today. And what is interesting here is that these macrocyanobacteria really refers to cyanobacteria that have big cell diameters. So they have, from 3 micron up to 50 microns. So these are huge things. And most of the microbial mats, marine microbial mats that we have today, have these, within this macrocyanobacteria group. I mean, these are 2 very broad groups, but what I think is really interesting is most of the diversity that we have today evolved after the great oxygenation event. Something, again, sort of relatively recent that we know is that planktonic groups, and I'm now talking about sort of marine ones, they evolve at the end of the Precambrian. So, and these are sort of important things in today's oceans, things like the ancestors of Synechococcus, Procolococcus, Trichodesmium. And I think you probably, your students have already sort of looked at some of these talks, nitrogen fixation. I think I looked sort of a while ago, not recently, some of the talks, but yeah, so these really important nitrogen fixers, primary producers in today's oceans evolved at the end of the Precambrian, and that has huge implications for obviously the biological pump, the amount of primary productivity, and food that would have been available as well for animals to evolve and diversified. And it's really until we get to that point that we have our modern Earth system. So yeah, I feel that there are still lots of things to discover about cyanobacteria in terms of those cyanobacteria that are important ecologically to link with, big biochemical cycles like nitrogen, oxygen, and carbon. This sort of late planktonic origin is important. Now, when it comes to, I think, the origin of photosynthesis as a whole, that's a far more complicated question. How are we doing with time? Yeah. That's a more difficult question, in part because we have cyanobacteria here. Let me see, I'm gonna make this small. We have cyanobacteria, and then we have other anoxygenic phototrophs. They use different electrode donors. Here we have the phylogeny of bacteria. And what is interesting, yet very puzzling, is that the phototrophs are all spread all over the tree of bacteria. So it's not that we have a monophyletic group. That makes the interpretation a lot harder. Some people have proposed that there is been lateral gene transfer. Oops, cannot go forward. Hang on. All right. Oh yeah. Oops. I think it kind of freaked out a little bit. Right. So this is work that so it's not published, so I won't say a lot of it, but some of this work is by my PhD student, now postdoc Giorgio Bianchini, and this is one of the figures from his work. So what we've trying to answer now is using the techniques that I mentioned, but using the bacteria tree with Bayesian statistical analysis to look at when in the bacteria tree photosynthesis evolve. But what I want to illustrate with this figure here, this is the bacteria phylogeny, and again, it sort of shows that we have all these different phototrophs all over the tree. Cyanobacteria has both reaction centers, type 1 and type 2, and the other ones have either type 1 or type 2. And this is, I guess, some people have postulated this lateral gene transfer also because people assume that because the early Earth was anoxic, there is an assumption that anoxygenic phototrophs evolved first. And I just would like to highlight that it's just an assumption, that needs to be tested and some evidence needs to be shown for that. But it's not easy to produce. I just want to highlight here that if you look at all phototrophs and we concentrate on the reaction center proteins, what is clear is that these reaction center proteins are part of a gene family and they have evolved through gene duplication. I'm going to take the example of the type 2. Type 2 we have here D1 and D2. D1 has the manganese cluster, which breaks water and obviously is uniquely representative of oxygenic phototrophs. You don't really need to know that much phylogeny to sort of see here that D1 and D2, which kind of work together, you'll find them together in the membrane, are more closely related to each other than, say, the anoxygenic phototroph ones, which is L and M. And that has implications for the lateral gene transfer hypothesis, because that implies the fact that they're more closely related to each other, that they duplicated within this lineage of oxygenic phototrophs. So there is no evidence for lateral gene transfer. With Tanay Cardona, we collaborated and we did, this was published in Geobiology 2019. If you do a phylogeny of these L, M, D1, and D2, and we have these ancestral sort of duplication events, what is clear is that these modern D1 appear closer to the great oxygenation event, and that the history of the gene family is older than the history of cyanobacteria, of those organisms that have left descendants. And I don't know if that makes sense, but what I kind of want to highlight here is that this really, the history of the gene, it's older than the history of those organisms that have left descendants. And I want to illustrate it here. So to the left, we have the species tree. So these are sort of all OEC cyanobacteria that we find today. And here we have these kind of, but this is obviously a cartoon, a diagram, but it's almost like the proteins likely evolve in organisms that have left no descendants. So they're sort of older than the cyanobacteria. Now, cyanobacteria are, their closest relatives are non-photosynthetic. They're part of the Cyanobacteria. This was proposed back in 2009. This has been confirmed recently. This is one phylogeny from a phylogenomic approach. Lots of different ribosomes and RNA, maybe about sort of 60. This is produced Tom Williams' group, and what is clear is that this Cyanobacteria is a well-supported bacterial group, and cyanobacteria sort of sit within that. And the name says it all, sort of land. So cyanobacteria sort of well sort of part of this good monophyletic group. Now, I think when it comes to estimating the age of cyanobacteria and their closest relatives, I think, different groups, kind of their molecular clocks are converging. This molecular clock comes from my group, my student Joe Bowden. We published that last year in Nature Comms, and it's about superoxidase dismutases, but I just want to highlight the divergence from the melanobacteria, these non-photosynthetic close relatives. This is a paper from Greg Fourier's at MIT, and we are sort of arriving from, to similar conclusions in terms of like a really deep divergence from these non-photosynthetic close relatives. And what I want to highlight here, this is a figure from Greg's paper from last year. We have the great oxygenation event. Here we have the Archean. We have basically this idea that, the deep branches of cyanobacteria are long, and the divergence from the close relatives, can be traced back to, 3.5, 3.7 billion years ago. So there is a long history, and what is very likely is we have these early phototrophs, and then we have another wave of cyanobacteria emerging before the GOE, and as I mentioned earlier, all this diversity after the GOE. So what's happening around the GOE is likely that we have all this, an expansion of filamentous forms, and contributing to this ecological dominance of cyanobacteria in land and coastal environments. With Joe, this is something that we have in PrEP, my student, where we're looking at the evolution of genes involved in multicellularity. Something else that I wanted to mention here is that you get this thing where you have this ancestral filamentous form, and then you have a lot of the loss of unicellular, the loss of filamentous, and things become unicellular. So that's another really interesting pattern. I did mention the end of the Precambrian, and we have all these sort of important primary producers. These are the nitrogen fixos, Trichodesmium, the Cyanobacteria Cocosphaera groups, and Xenococcus and Procolococcus. So you can imagine that, you know, these vast areas of the ocean started to be colonized by these ancestral planktonic groups, and that would have helped transform the Earth system, primary productivity. This is a paper from 2014 where we have this the age for these different groups with molybdenum. Molybdenum is only soluble in oxygenated waters, and that when we first proposed this late planktonic origin of cyanobacteria. Similar, this is a paper that I have on the origin of the chloroplast. So happening sort of at around 2 billion. Glowing margarita is the closest cyanobacteria. But what is interesting as well here, if you look at, these are green algae, and we have land plants, green algae, red algae, that I'm sort of pointing out here. But these 7 and 8, these are the first planktonic green algae. And I find that really interesting because, again, we have this sort of radiation of other planktonic groups. It's likely that they would have needed the nitrogen that cyanobacteria were fixing, and a lot of different things. It's a very dynamic part from the perspective of primary producers, and all of, you know, and there is a lot of sort of geochemical evidence sort of showing lots of things that are happening with the carbon nitrogen cycle. And, you know, all this sort of green algae, there is a lot of evidence from sort of biomarker data that they're definitely appearing around this time. Oops, I thought I had deleted this slide. I wasn't going to talk about that. And that's it. That's a very brief, you know, very hopefully quick version, but yeah, 31 minutes. So